Hello my dear students, this is Dr. Pooja, your biochemistry mentor. Now in this video, we are going to discuss about a very interesting amino acid which is known as the simplest amino acid, optically inactive, non-essential, but it is again required by the body to form so many specialized products, and this is glycine. So now we are going to discuss the metabolism of glycine. Now first of all, let's look at the structure, how glycine looks like. This is the generalized structure of an amino acid, obviously, which is same for all the 20 standard amino acid, and here H is present. So this becomes the structure of glycine, where you can see that this C or the entire structure lacks a chiral carbon atom. Right, so since it is lacking a chiral carbon atom, so we can call it as asymmetric also. Now, since it is asymmetric, it is optically inactive also, because we all know that if a molecule lacks a chiral carbon atom, it is always going to form a superimposable mirror image, or it is not going to rotate the plane of polarized light in any of the directions. So we can say it is optically inactive, right? And it is also a non-essential amino acid. That means it can be easily synthesized in the body. Coming over to the metabolism as per carbon skeleton, it is a glucogenic amino acid. Right. So glycine, we can say it lacks a chiral carbon atom, so it is asymmetric. Since it lacks a chiral carbon atom, it will be optically inactive, it is non-essential, it is glucogenic, and it is polar in nature. Right? Polar, but you can say uncharged. Now the question is, first of all, since it is a non-essential amino acid, so how it is synthesized in the body? And what are the specialized products synthesized by glycine? So, if we talk about glycine, I can simply say that there is one function as catabolic. That's how we are studying all the amino acids. And another is the anabolic role. Now, if we talk about the catabolic role, we can categorize glycine into two categories. One is there is interconversion of glycine to serine, and there is a glycine cleavage system. And if we talk about just the anabolic nature of glycine, it is required for the synthesis of purines. It is required in the synthesis of heme. Then it is required for the synthesis of creatine, and it is required for the synthesis of glutathione. Then obviously it helps in conjugation with the bile acids. So this is the summary of the metabolism of glycine. So step by step we are going to study. Now this glycine is actually, if we talk about the synthesis of this, we can simply say that glycine can be synthesized from threonine, by the enzyme threonine aldolase, right? Then this glycine can be converted into serine, by the enzyme hydroxymethyltransferase. Right, and you can say here glycine synthase, which will require folic acid for its function. Right, and similarly glycine can be converted into glyoxylate. Right. Now the question is that this glycine will also tend to undergo transamination being an amino acid. So how this glycine is converted into glyoxylate, because this is a very important reaction. Second is how glycine can undergo cleavage by this glycine cleavage system. And if we talk about glycine to serine, we can simply say that glycine synthase, because synthesis is there, and what we have to put is hydroxymethyltransferase, and this methyl group is coming from folic acid. Clear? Now the question is, this glycine, first reaction, can be converted into glyoxylate by the enzyme 𝗖𝗵𝗵𝗿𝗺. The name of the enzyme here is glyoxylate alanine aminotransferase. Right. Then glycine, being an amino acid, will also undergo transamination. So we can say that transamination is simple: glycine plus alpha-ketoglutarate will form glutamate and glyoxylate. And we all know that transamination is a reversible process, and the reaction enzyme is obviously aminotransferase, and the coenzyme would be P6. This is a very important reaction, that how glycine is converted into glyoxylate, taking pyruvate and alanine into this, because absence of this enzyme will lead to hyperoxaluria, which we are going to discuss later. Then second reaction is, what do you mean by this glycine cleavage system? Glycine cleavage system. Now what happens is, it has 3 enzyme activities at the same place. Now what happens is 1, 2 and 3, third enzyme. Now first enzyme is COOH and H2. Right. First enzyme would be taking of this CO2 group. So this activity is known as simple. This is going, so we can say that it is dehydrogenase or decarboxylase. Then removal of this amino group, so it will be simple aminotransferase. We can say glyoxate or glycine, we can write aminotransferase. Right, and third is dihydrolipoamide dehydrogenase. Right, and we can simply say that there will be conversion of THF to N5N10 methylene THF, or I'll just write methylene THF. so everything can go out, right? So this is the, you can say, glycine cleavage system. Then another reaction is, now what are the specialized products which are synthesized from glycine? Very, very important. So specialized products from glycine. Now if we talk about the first one is heme synthesis. So always remember that glycine is required for the synthesis of heme, and we all know that heme synthesis takes place in two compartments, mitochondria and the cytoplasm, right? So if we talk about the first reaction is when succinyl coenzyme A combines with glycine and it forms ALA, which stands for amino levulinic acid or levulin. And the name of the enzyme here is ALA synthase, which requires B6 for its activity. But the most important thing is glycine. And this is a rate limiting enzyme also. This is the rate limiting enzyme of heme synthesis. Right? So this is the first stabilized product, or you can say specialized, because whenever there is increased synthesis of heme, there are so many steps, so I can say whenever heme ka concentration increases, it inhibits this enzyme. And this enzyme is requiring succinyl coenzyme A plus glycine. So this is first very important feature of glycine. Second is glycine is required for the synthesis of glutathione. Synthesis of glutathione. Now, if we talk about glutathione, we all know it is a tripeptide. Glutathione is a tripeptide which is formed of 3 amino acids, glutamic acid or glutamate plus cysteine plus glycine. Now, if we talk about this, we know what happens is this is also known as gamma glutamyl cysteine glycine. Right, this is the other name of this, and it is an example of pseudopeptide. It is an example of pseudopeptide. Again, this is very important. So what happens is glutamic acid it will be combining with the cysteine, and then glycine will come, giving it a very important, you can say a very important compound is produced. And if we talk about the functions of glutathione, there are innumerous functions of glutathione. We know that if we talk about the functions, we know that glutathione is a very, very important antioxidant. It is an antioxidant. And from where this antioxidant nature is coming? It is coming from the SH group of cysteine. Right? Second is glutathione is required for the transport of amino acids. So it is required for the transport of amino acids by a very important cycle which is known as Miester cycle. This is also a very important feature of glutathione. Then third is it helps in maintaining, it maintains the integrity of RBC's membrane. Very important. Cell membrane. Right. So these are the three very, very important functions of glutathione. Now, if we talk about this, if we elaborate this function, glutathione occurs in two forms, oxidized and reduced. Right. So oxidized is converted into reduced with the help of NADPH, which is converted into NADP positive. Right. And here if we talk about the enzyme here is glutathione reductase, which requires B2 for its activity. Right. And if we talk about this reduced form, whenever glutathione is in its reduced form, it is going to break hydrogen peroxide into water and oxygen. Right. And here the enzyme is glutathione reductase. And here it is glutathione peroxidase, which requires selenium for its activity. So we can say that two enzymes are there, glutathione reductase, glutathione peroxidase, but glutathione is the one which is playing a major role and glycine is required for this. And absence of the enzyme catalyzing the entire reaction leads to hemolytic anemia. Right? So this is another function, and glutathione is again having glycine. Then third function of glycine is synthesis of creatine. Synthesis of, and what is creatine? Creatine is the one which is the energy reservoir of the muscles. Energy reservoir of muscles. Now, what happens is the synthesis of creatine takes place in two organs. So, what happens is that first of all, it is a tripeptide. And what are the three amino acids required for the synthesis of creatine? It is glycine, arginine and methionine. So what happens is this glycine combines with arginine, and this reaction takes place in the kidneys where they are going to form guanidoacetate. Right. So what happens is this guanidoacetate then combines with methionine. All the reactions are stepwise. First of all, glycine will combine with arginine in the kidneys to form guanidoacetate, and this guanidoacetate then will combine with methionine to form creatine. And this reaction takes place in the liver. So basically the most important reaction, that is methylation, transmethylation, takes place in the liver forming creatine. And this creatine can be converted into creatine phosphate, obviously to give energy to the muscle, and this reaction takes place in the presence of CPK enzyme using ATP. It is known as Lohmann's reaction. On the other hand, this creatine loses a molecule of water minus water and it is converted into creatinine. And we all know that creatinine clearance test is a very important component of renal function tests. Right. So you can see again, if we talk about the importance of glycine here, you can see that glycine is there. So this glycine is the one which is leading to the formation of, we can say creatine or we can say creatinine also. Right. So this is another important feature of glycine. Then we come over to the fourth function. Glycine is required for the synthesis of C4, C5 and N7 of purine nucleus. Right, and we all know if we talk about purines, purines have an extra imidazole ring like this. Numbering starts like this: 1, 2, 3, 4, 5, 6, 7, 8 and 9. So C5 and N7, this chunk comes from glycine. This is also a very important feature of glycine, that C4, C5, in fact major backbone of purines is coming from glycine. So these are the important functions, and along with these other functions are that glycine is required for the synthesis of bile salts or bile acids. So synthesis of bile acids or bile salts. Right, so these are some of the specialized products synthesized from glycine. So this is the anabolic and catabolic nature of glycine. Then after this we come over to what are the inborn errors of metabolism of glycine. So if we talk about the inborn errors of metabolism of glycine, the most important one is hyperoxaluria. And what happens? It is primary, it is secondary. Now, if we talk about primary oxaluria, what happens here is, it is due to the deficiency of enzyme. The deficient enzyme here is glyoxylate alanine aminotransferase. And if we talk about secondary hyperoxaluria, here the deficient enzyme is glyoxylate reductase. Right? And both will lead to oxaluria. So these are the inborn error of metabolism associated with glycine. Right? So if we have to just summarize the entire metabolism of glycine, we can simply say that it is non-essential, very, very important. It is optically inactive. Then glycine is required for the synthesis of heme, for the synthesis of purine nucleus, C4, C5 and N7, and it is required for the synthesis of We can say bile salts, or we can write creatine. We can write glutathione. And glycine can be converted into serine. So this is how we can correlate the metabolism of glycine and how glycine is being produced. It is being produced from threonine in the presence of enzyme threonine aldolase. Right. So this is all about the metabolism of glycine, which is a non-essential, simplest amino acid which lacks a chiral carbon atom and which is optically inactive. I hope you have understood this video. Thank you so much.